Hello and welcome to my channel. In this lecture we will discuss the auditory pathway. So let's begin. The auditory system is connected with the vestibular system concerned with balance because both of them carried by one nerve, the eighth cranial nerve, which is the vestibulocochlear nerve. So the receptors are different. Receptors, of course, there are the auditory organs and the vestibular nuclei that project to the brain as the vestibulocochlear nerve. The auditory pathway, this is a cut section, coronal section of the ear. This is the external ear till the drum, and then from the drum till the cochlea, this is the middle ear, containing the ossicles, and here's the internal ear, the bony labyrinth, which is the cochlea and the semicircular canal comprising the inner ear, of course looking from anterior aspect. Sound waves, they are mechanical stimuli. They are collected by the ear pinna. In lower animals, the external auricular muscles are well developed more than human beings. Human beings has remnants of these muscles like auricularis superior, anterior, and posterior, but it collects the sound waves by the ear pinna and concentrates them into the external auditory canal, directing them to the drum, or the tympanic membrane. The drum response is characterized by faithful response, means that it doesn't modify the sound wave, neither add or take from it, from the stimulus or from the wave coming and hitting the tympanic membrane. It's called the faithful response. Also, it's characterized by critical damping. It damps harmful frequencies. So it has faithful response and critical damping. It damps harmful frequencies. And it conveys sound waves to the ossicles, as you can see here. These are the ossicles, the malleus, the incus. This is the malleus, this is the incus, and this is the stapes. In the middle ear, and amplification of the sound takes place. Now, don't think that the ossicles are the major component or the major factor for amplification of sound waves. No, it's the sound amplification is achieved by first of all the ratio between the tympanic membrane, the surface area of the tympanic membrane, compared to the surface area of the oval window. Tympanic membrane, oval window. This achieves around 17 times amplification of the sound waves. Also, the ossicles share in the amplification by a factor of 1.3 times, so the net amplification will be 17 times 1.3. The sound is amplified 22 times before reaching the oval window. Attenuation or weakening occurs due to actions of the tensor tympani and stapedius muscles. Remember, the stapedius muscle attached to the stapes and supplied by the facial nerve, and the tensor tympani muscle. We studied this in the middle ear. The tensor tympani is supplied by the nerve to medial pterygoid from the mandibular of trigeminal, and this attenuation helps in protection of the inner ear, decrease in the background noise, and decrease in the hearing self voice. So attenuation is important for protection and decrease in the background noise and decrease of hearing self voice. In the inner ear, this is a cut section of half a turn of the inner ear of the cochlea. The cochlea is 2.5 turns, turn, another turn, and a half. So the cut section shows half a turn of the cochlea, where it has 3 scala: the scala vestibuli, the scala media, and scala tympani. The scala vestibuli is separated from the media by the vestibular membrane, and the scala media is separated from the tympani by the basilar membrane that has the organ of Corti on it. See, all this is the organ of Corti, composed of hair cells and the tectorial membrane. And the scala vestibuli and scala tympani are filled with perilymph, more or less like the plasma composition. But the scala media is filled with endolymph, more in composition or in relation like the intracellular fluid. It's produced by this region, the stria vascularis, at the side of the scala media. The vestibular membrane has no resistance to sound waves, and receptors are the outer hair cells and the inner hair cells. Outer hair cells and the inner hair cells. Outer hair cells have contractile proteins in them, so they affect the basilar membrane, and outer hair cells more than the inner hair cells, which share in the response to sound waves. Receptors, the hair cells, I mean, they receive olivocochlear fibers coming from the superior olivary nucleus, and the receptors may be damaged by age, noise, hypoxia, drugs like certain antibiotics, for example, and genetic factors. There are also phalangeal and pillar cells, for support, and at this level, transduction of mechanical sound means the conversion of the mechanical sound wave into an electric impulse occurs at this stage. So the sound wave travels through the scala vestibuli, vibrates the membrane, and vibrates the scala media, which vibrates the organ of Corti, and the rest of the impulse travels through the scala tympani and hits the round window to be terminated, and the receptors are ready to receive another mechanical impulse. Now, transduction, regarding transduction and frequency coding, there is what's so-called the place principle. The place principle is where every part of the basilar membrane responds to certain frequencies, means it differentiates the frequencies or separates the frequencies, or all over the membrane, the basilar membrane, each point in the basilar membrane responds to a certain frequency. The basilar membrane is widest and least stiff at the apex of the cochlea, and narrowest and stiffest at the base or near the round and oval windows. This means that the high frequency sounds localize near the base of the cochlea, where the membrane is stiffest, and the low frequency sounds localize near the apex, where the membrane is least stiff and widest. Okay, so remember that. It's high frequency sound localized near the base of the cochlea, and low frequency sounds localized near the apex of the cochlea. Sensory receptors then synapse with sensory neurons, as you can see here, which have cell bodies in the spiral ganglion. Remember the spiral ganglia we studied in the modulus of the cochlea, and converts the modulus into bony fragments, actually, because it occupies the modulus. Then the cochlear nerve is formed. The cochlear nerve is the axons leaving the modulus from near the base of the cochlea, and the nerve leaves through the internal acoustic meatus to the cerebellopontine angle. So transduction now is complete. The mechanical impulse is converted into an electrical impulse, and remember that the cochlear nerve is the axons of the cells of the spiral ganglia in the modulus. Now the central auditory pathway begins. The first order axons terminate ipsilateral on the dorsal and ventral nuclei, cochlear nuclei in the medulla oblongata. This is a cut section of the medulla oblongata. Second order axons ascend in the auditory pathway, as you can see here, and projects bilaterally on the superior olivary nuclei. The superior olivary nuclei, when studied their function, they are important in determining or in localizing the origin of sound, determining the direction of sound, where it comes from. Fibers continue to ascend as the lateral lemniscus, this is the lateral lemniscus here, to the midbrain, where it terminates in the inferior colliculus. Remember the inferior colliculi, we said they are part, pardon me, they are part of the central auditory pathway. Now fibers leaving the colliculi go through the inferior brachium to the medial geniculate nucleus of the thalamus before synapsing in the auditory cortex or primary auditory cortex in the superior temporal gyrus of the temporal lobe. There is also a pattern which is called the tonotopic representation. So tonotopic means the cortex has one end for low frequencies and the other end for high frequencies. The superior olivary nucleus receives feedback from the cortex, cortico-olivary fibers, then sends inhibitory fibers to the organ of Corti to protect them from damage from loud sounds. The superior olivary nuclei also have connections with the motor nuclei of the trigeminal and facial nerves. It makes sense because these nerves control the stapedius and tensor tympani muscles inside the middle ear for further protection of the inner ear from the loud sounds. Okay. Vestibular pathways. Vestibular nerve transmits information regarding the movement and position of the head from the semicircular canals or the semicircular ducts, to be precise, and utricle and saccule. We studied this in the inner ear, the arrangement of the inner ear. The cell bodies of this for sensory receptors located in the vestibular ganglion. It's called the ganglion of Scarpa, within the internal acoustic meatus. And the receptors are the crista ampullaris of the ampulla. This is the crista ampullaris in one of the ampulla of the semicircular ducts. And the crista ampullaris is responsible for detection of angular acceleration and deceleration movement. The receptor in the utricle and saccule, called the macula, it's of different shape than the crista ampullaris, but the same principle of action. And it detects movement and position in the vertical plane. So remember, the crista ampullaris for angular acceleration and the macula for vertical movement in the vertical plane. Vestibular nerve then leads to the cerebellopontine angle together with the cochlear nerve. Now the nerve is complete. The vestibulocochlear nerve, the eighth auditory nerve, lies lateral to the facial nerve at the cerebellopontine angle at the lower border of the pons. And this cerebellopontine angle, we mentioned before that it's common site for tumors like meningiomas, or for example, that may affect hearing and balance by affecting the vestibulocochlear nerve at this location. Now this is a plate of the internal ear. See, this is the cochlear duct, and this is the utricle and saccule and the vestibule. And the semicircular ducts, they occupy the bony labyrinth, which are the semicircular canals, the vestibule, and the cochlea. So note to differentiate between the bony labyrinth and the membranous labyrinth. Membranous labyrinth are what we just said, the semicircular ducts, the utricle, saccule, and the cochlear duct in the cochlea. Of course, this region is responsible for hearing, and this region is responsible for balance. Okay? Now, after forming the vestibulocochlear nerve, the central vestibular processes terminate on the four vestibular nuclei we studied before: the superior, inferior, lateral, and medial vestibular nuclei. In the medulla and lower border of the pons. Axons leaving the nuclei connect with the visual, motor, descending motor, and cerebellar pathways to coordinate movements and maintain posture and balance. Also, axons leaving the medial vestibular nucleus going upwards through the brainstem, upwards and downwards to cover the brainstem. These fibers are called the MLF, the medial longitudinal fasciculus, that connects the cranial nerve nuclei of oculomotor, trochlear, and abducens nuclei together for visual fixation on the object. Also, other fibers from the vestibular nuclei project to the cerebral cortex after relaying in the ventral posterior nucleus of the thalamus, ventral posterior nucleus of the thalamus. This is for orientation in space. Other fibers enter the inferior cerebellar peduncle to cerebellum to modulate the equilibrium. We studied this with the cerebellum. Now, axons descending in the medial and lateral vestibulospinal tracts to the medial motor system for control or modifying the action of the axial muscles, mainly in the spinal cord. This is achieved for posture and balance. So, as you can see here, the vestibular nuclei and the MLF is forming with the lateral vestibulospinal and medial vestibulospinal tract. And please note that the connection with the cranial nerve nuclei inside the brainstem, as we mentioned before, for visual fixation on the object. Well, this concludes our lecture. I hope you got the maximum benefit from this lecture, and thank you very much for watching.